In 2003, a virus known as SARS coronavirus emerged from bats into the human population. It infected over 8,000 people with a 9% mortality rate before the virus was eliminated. In 2012, a closely related virus known as Middle East Respiratory Syndrome, or MERS coronavirus, also jumped from bats into humans. To date, MERS has infected over 1,500 people with a 36% mortality rate and is still circulating in the population. Unfortunately, there are no drugs or vaccines available for either of these viruses. The current threat that MERS poses makes the development of potential therapeutics a high priority. However, while MERS can infect humans and bats, it cannot infect any of the small animals that are typically used to study infection, such as ferrets, guinea pigs, hamsters, or the most common small animal model, the mouse. Without an animal model for MERS, we cannot begin to study how the virus causes disease or begin to develop drugs or vaccines. 7 years ago, I was a nascent undergraduate working in an epidemiology lab. I was at a dinner party with my advisor and several professors when they began talking about some complicated science I couldn't even begin to follow. However, at one point my advisor said, "You know the solution to your problem, don't you? You make a mouse." Everyone in the group chuckled, and I chuckled as well, because of the absurdity of the statement, but also to make it seem like I even remotely knew what they were talking about. 5 years later, I'm a nascent graduate student working in a coronavirus lab. When MERS emerged, my advisor approached me and said, "Figure out why this virus can't infect mice." I, naturally, gave him an overeager "yes, sir." But as he was walking away, I called out after him, "But why?" He turned to me and said, "So we could make a mouse." I chuckled, as I had done 5 years before, and it wasn't until he stood there staring at me that I stopped and realized he was serious. My thesis focuses on explaining why MERS can infect humans and bats, but not mice. We have found that the answer lies at the level of the host cell receptor, or the protein that the virus interacts with in order to enter and infect a cell. In fact, we found that with just 2 mutations to the mouse cell receptor, MERS can easily infect a mouse cell. Using this information, our lab is doing what at one point in my life seemed like pure science fiction. We are going to make a mouse. With the development of a transgenic mouse that can be infected by MERS, researchers around the world can begin to study how the virus causes disease. This knowledge can not only help in the treatment of those infected with MERS today, but can better prepare us for future coronaviruses that might jump into humans. While we cannot predict if or when the next coronavirus will emerge, at least we know that to fight it, we can always make a mouse. Thank you.